So hi everybody, welcome to today's webinar. So it's going to be on uncovering protein function with UniProt. So my name is Michelle McGrain and I coordinate a team of curators at EMBL-EBI who contribute to the information content of the UniProt resource. And today my talk will be in two parts. So in the first part of the talk, I will describe how information is added to the resource, where the information comes from. And then in the second part of the talk, I will show you how you can find functional information on the UniProt website. So to start with, what is UniProt? So UniProt is a resource of protein sequence and functional information, and it aims to provide comprehensive and high quality data, and all of the information is made freely available through the website uniprot.org, and the data is released every 8 weeks. And the resource is produced by the UniProt consortium, which is made up of groups at EMBL-EBI in the UK, at the Swiss Institute of Bioinformatics in Geneva, and the Protein Information Resource at Georgetown University and the University of Delaware in the US. And these three groups together work closely together to maintain the resource. So in UniProt, UniProt is a sequence resource, and the sequence data are organized into different resources. So we have UniPark, which is a sequence archive of non-redundant, publicly available sequences from different databases. UniProt also provides UniRefs, which are protein sequences which are clustered at various sequence identity levels. There's access provided to proteins which are part of complete proteomes through the UniProt Proteomes portal. But the centerpiece of the activities of the consortium is production of the UniProt knowledge base, which acts as a central hub for functional information on proteins and provides also access to their sequences. And so my talk today will focus on production of the UniProt knowledge base. So why use UniProt? Well, we all know that the amounts of data that are being generated continue to increase, and it's a really difficult task to stay on top of all this huge amount of information that is being constantly produced. And so the UniProt knowledge base helps researchers to access this information and organizes the information. So it aims to provide comprehensive and high quality data. And it also helps researchers and aids scientific discovery by collecting, interpreting and organizing huge volumes of data. And this saves researchers time in having to go and look for this information and collect all of this information for themselves. And as well as all of the data that is provided through UniProt KB, there are also tools provided which can help with protein sequence analysis. So things like tools for BLAST or aligning protein sequences. And UniProt also links to a very large number of other resources. So there are cross-references provided to over 180 other databases. And so this means that you can use UniProt knowledge base as a starting point and then jump out from a particular protein of interest to explore the information about that protein in other resources. And there are two data sets in the knowledge base. So there is the reviewed SwissProt section and the unreviewed Tremble section. So in the reviewed section, all of the data are manually reviewed. And the data comes mainly from scientific literature, and also that's coupled with manually reviewed results from computational sequence analysis. And in contrast, the unreviewed Tremble section has no manual review of the data. So the protein sequences are imported from various sequence repositories, and then additional data is generated from computational pipelines. So where do the sequences in the knowledge base come from? So most of the sequences come from translations which are contained in submissions of nucleotide sequences to the databases of the INSDC, which is the International Nucleotide Sequence Database Collaboration, which comprises the European Nucleotide Archive, or ENA, which is hosted at EMBL-EBI, GenBank at the NCBI, and the DNA Data Bank of Japan. And if any submissions to those resources contain coding sequences, then those protein translations are automatically imported into the unreviewed Tremble section. And in addition, we also import predictions from various genome annotation resources such as Ensemble and RefSeq. So these resources, so many genomes are submitted without any annotation. So then these resources will take those genomes and, excuse me, run them through their pipelines and to predict regions of interest within the genome, including protein coding regions. And so those proteins can also be imported into the knowledge base. We also have a pipeline to import sequences from the protein data bank so that we can ensure that for every protein which has a structure in PDB, we have a corresponding entry in the knowledge base. And we also accept submissions directly to UniProt of directly sequenced proteins. And so this is a very small proportion of the total number of sequences in the knowledge base, but it's an important source because many of these directly sequenced proteins also have characterization data associated with them, which allows us to provide information about the function of these proteins. And then unreviewed Tremble entries may go through the expert curation process to become reviewed entries within the SwissProt section of the knowledge base. So on the next few slides, I'm just going to talk you through what is involved in the manual curation process. So UniProt knowledge base is a protein sequence database, and although we add many layers of data around the sequence, we want to make sure that the sequence that we provide for each protein is as accurate as possible. The SwissProt reviewed section is non-redundant, so that means that sequence reports that are coming from the same gene and the same organism will be merged into a single SwissProt record. We also try to ensure that these sequences match the genome, and curators can correct protein sequences if necessary. So if there are any discrepancies that look like they may come from things like sequencing errors, then the curators will go in and actually manually correct the sequence. And any differences in sequencing reports from what is shown as the main displayed sequence are also documented. And this can be due to things like I've already mentioned, like sequencing errors, but also naturally occurring events, so things like natural variants or isoforms arising from things like alternative splicing. And so this can be a time-consuming step, but it makes sure that the sequences that we provide in SwissProt are as accurate as possible, and so that then ensures that any further sequence analysis gives as accurate results as possible, and it also can feed back to upstream genome resources like Ensemble and RefSeq to aid in improving future predictions of proteins. And then once the sequence is acceptable and correct, then the curators will run various sequence analysis programs, and these are run to predict things like post-translational modifications, transmembrane domains, and other domains of interest. And all of the predictions are manually reviewed, and only relevant results will be selected for inclusion in a record. And then these predicted results are combined with experimental data from the scientific literature to provide a complete overview of the sequence features for a particular protein. And most of the information that's added during the manual curation process comes from the scientific literature. So the curators read a huge number of publications and extract data from these papers to help explain the function of a protein, and then take that unstructured data and put it into a UniProt record, organize the data, use controlled vocabularies. And in the second part of the talk, I will show how this kind of information is shown on the website. So during the literature curation process, experimental findings will be critically evaluated. They will be compiled into a comprehensive report. The information is represented, as I said, using controlled vocabularies wherever possible. And all of the information is linked back to the original publication so that it's clear which paper is the source of a particular piece of information. And so literature curation is really valuable because it ensures that published results in the scientific literature are extracted and converted into a format which can both be easily understood by users who want to come and read a UniProt record, but this information is also computationally accessible. And data from a particular paper can be combined with information from other papers and from other sources to give a complete overview of the role of a protein. And also curators will highlight any conflicting or erroneous data and make that clear to users as well. So this process, in the end, provides a complete overview of a particular research area. So how do we actually find this literature? So we have a number of ways to do this. So we monitor new publications that come out in a set of high impact journals. We also make use of what we call our additional bibliography, and these are papers that come from other resources and that are computationally mapped to records within the UniProt knowledge base. We, of course, make use of literature databases. We have a very active user community who will send information about papers which are missing from the resource. And we also have a facility to allow community submitted literature, and we make use of text mining tools as well to help with identifying relevant papers. So I'll just say a little bit about some of the text mining tools that we're using. So we use, one of the tools we use is PubTator. So this is a freely available tool from the NCBI, and we're making use of this for weekly literature triage. So we can run this over a set of high impact journals which we're interested in. So journals like Science, Nature, Cell, and others. And this allows us to keep up with what the high impact papers that are coming out on a weekly basis. Then we also make use of the LitSuggest tool. So LitSuggest, again, is another freely available tool from the NCBI. And this is very useful for helping to keep track of the literature associated with a particular area. So within UniProt, we have been using it successfully to find papers dealing with disease-causing variants and papers reporting new enzymatic reactions. And so the tool allows you to train a model, and then once you have the model trained, you can set it up to run on a regular basis, and it will send an email report with papers of interest in your particular area that you're looking for papers in. We're also exploring the use of large language models to help with identifying and reviewing relevant literature. So obviously large language models are likely to be very helpful in this field. in things like scanning and summarizing published papers, also in helping to identify papers which could add new information, and also in tasks like being used as a co-pilot for curators to help with writing scientific summaries by helping to integrate information from a number of papers. So we're currently running various pilot projects to assess how LLMs can help with these tasks going forward. Something else which we make use of is crowdsourcing the literature. So this really helps us then to make use of the scientific knowledge of the research community. And and this is a kind of a two-way process. So it allows researchers to play a role in contributing to the content of UniProt, and obviously while also being very beneficial for UniProt in collecting new papers. And researchers who contribute in this way do receive attribution for their contributions, and I'll show a little bit more about this in the second part of the talk about how this is shown on the website. And obviously, like I say, this is a two-way thing, because an improved database then better supports the entire research ecosystem and helps to advance science. So once we have identified information from the literature, then curators read a lot of papers and they extract a huge amount of information from these papers. So everything from protein and gene names through to information about the function of a protein, for enzymes, what reactions they catalyze, and if they use any cofactors, where a protein is located within a cell and in what tissues it's expressed. Does it require interaction with other proteins for its function? And then there's also a lot of information captured from the paper at the level of the protein sequence. So things like splice variants of a particular protein, any domains or regions of interest within the protein, important binding sites, post-translational modifications, and residues, which are important for catalysis. And curators take all of this information and they really add value to it by collecting it, compiling it together into a report, organizing it and representing it using controlled vocabularies. And so they produce a report, like I say, that can be easily read by users, but also that can be accessed computationally. So manual curation is really the gold standard, and it's really essential for producing very high quality data. But it doesn't really scale in the face of the huge amounts of sequence data that continue to be produced. So this slide shows how Tremble is continuing to grow. So it now has so this is the unreviewed section, has now over 250 million records. So although manual curation is the gold standard, we obviously need ways of improving the data within the unreviewed section. So these are the entries that haven't gone through the manual review process. And so to do that, we have various automatic processes which we use. So there is some automated sequence annotation done. So there are various sequence analysis programs run over the unreviewed records in the Tremble section to predict signal sequences, transmembrane regions, coiled coils, and intrinsically disordered regions. And then domains are also predicted based on the member databases of the Interpro resource, which is a resource that is like an umbrella database that brings together various databases which produce signatures to detect important domains, regions, and sites within proteins. And so those domains that can then be added to the unreviewed Tremble records. And in addition, we have a number of rule-based systems to provide automatic annotation to the unreviewed records. So then we have a UniRule system where all of the rules are, so although the system is applied automatically, the rules themselves are manually created by curators. and the ARVA system, which is completely automated and uses rule mining. And what these systems have in common is that they make use of interpro to group proteins together that belong to a particular protein family, and then they extract the common annotation from those from the manually reviewed entries within that set, which can then be transferred to the unreviewed records. So just to show in a bit more detail how this works. So on the left-hand side here, this just shows a set of reviewed entries. So these are bacterial entries, which are all part of a particular protein family, which is defined by HAMAP, which is one of the Interpro member databases. And these reviewed entries share common annotations, so things like the protein names, the function that the protein carries out. It's an enzyme, so they all have the same EC number. And so a rule has been manually created based on this information to say that if a protein is part of this family, which is defined by this HAMAP signature, then these annotations can be added to any of the unreviewed records which are part of that family. And this then really allows us to go from doing functional characterization from the literature to really doing annotation at scale. So for example, this rule provides information for 15,000 unreviewed records in the Tremble section of the knowledge base. And you can see that this is an unreviewed record, but it contains a lot of information, even though it hasn't been looked at by a curator. So this system is very powerful in providing a lot of information, even for records which haven't been experimentally characterized or gone through the manual review process. And the rules are also made available for anyone who wants, as a standalone version, for anyone who wants to annotate their own sequences using Unifier, which is an engine that executes the rules. So, and I've put the link to the Unifier on the slide for anybody who's interested. And we have also collaborated with Google Research to develop a system called PROT-NLM, which is a natural language processing model, which is currently being used to name proteins which previously didn't have any meaningful protein names. So these were just called uncharacterized proteins. And so this system can provide a meaningful name, which is very useful because that's something that people often see in things like blast searches. So again, if this is something of interest, I've put a link on the slide here, which gives more information about the model, and there's also so how it works, and there's also a link to a preprint of a paper describing the system as well. And so yeah, this is providing names for a very large number of records within the unreviewed section. So yeah, provides names for about 30 million proteins, and altogether all of the automatic annotation systems together provide some kind of information for around 90% of the records in the unreviewed section. And if you're interested in learning more about automatic annotation in UniProt, we will be doing a webinar at the end of January. So yeah, it'll be announced on the EBI training. So just keep an eye out for that if that's something that you're interested in. And as well as all of the information that's added directly, there's a large amount of information which is pulled in from other resources. So a number of large-scale data sets. So there's a large-scale variation data pulled in from multiple resources, large-scale mutagenesis data, which comes from the INTACT database. Information on post-translational modifications comes from Proteome Exchange. We also import proteomics identifications from a number of large-scale mass spectrometry repositories, and we also include AlphaFold predictions. So all of this information is added on top of what's coming from the literature and the automatic annotation systems. So as you can see, there's the information and the resources coming from lots of different sources. So from things like scientific literature, from researchers, from other databases, from various computational pipelines. And so it's really important that we provide a way for people to know where all of this information is coming from. And so all of the information in the knowledge base is linked back to its original source. And to do this, we use the evidence and conclusion ontology, or ECO. And this is an ontology that was designed to provide information about the support there is for a particular biological assertion. So, for example, if a paper shows that a protein, it reports experimental data showing that a protein acts as a tyrosine kinase, then a curator can add that information to the UniProt record and use ECO to show that the information is the source of the information, and also that the information is experimental. So using this evidence system, users can tell exactly where each piece of information has come from. They can tell whether the information is experimental or predicted. If it's experimental data, they can know which paper it comes from. And if it's predicted data, they can tell which system has been used to predict the information. So just to summarize the first part of the talk, so the UniProt knowledge base is divided into two sections. There's the reviewed SwissProt section with just over 500,000 expertly reviewed entries, where the information comes primarily from scientific literature, and with information from results from manually reviewed results from sequence analysis tools. So all of the data in SwissProt is manually reviewed. And then there's the unreviewed Tremble section, which is much larger. There's over 250 million records in the Tremble section. The information comes from UniRule and ARBA pipelines, and also from automated sequence analysis. And all of the data in both sections is linked back to its original source. So I'm going to move on to the second part of the talk now to show you where you can find this information in UniProt. So this shows the UniProt website, uniprot.org, and there is a search bar on the homepage. You can do simple queries. You can also do advanced queries. And so once you've found your protein of interest, you get taken to an entry page, which looks like this. There's a menu at the top, so you can use this to choose how you want to view the entry. And there is also a number of quick access tabs along the left side of the page, and that allows you to jump between specific sections so that you can easily find the information that you're looking for. So, for example, if we want to have a look at the functional information for this protein, we'll click on the function section in the menu, and we're taken to the function section. So this gives so there's free text information about the protein, which has been compiled from multiple papers. There's for enzymes, reaction details are provided, and this is done using RIA, which is a database of enzymatic reactions. And all of the information, as I said, is linked back to the source publication so that you can see exactly where the information has come from. So as I said, this is so the protein that I was showing here is called myotubularin 2. It's a lipid phosphatase. So in this case, it has catalytic activity. You can, within the catalytic activity section, you can click on a reaction participant to access further information. And if you want to see which papers are supporting a particular piece of information, you can click on the publication tab that is associated with that piece of information and expand that, and it will give you the details of the paper in which that piece of information was reported. And as well as the free text description of the function, there are gene ontology terms provided. So UniProt curators assign gene ontology terms to entries when they are curating them. So, so if for anyone who's not aware of the gene ontology, it's a controlled vocabulary to describe molecular functions of a protein, in which biological processes they're active, and where in the cell they're located. So you're provided with this high-level overview, which just shows high-level terms, so you can get just a very quick overview of what functions, processes, and cellular locations for this particular protein. And then there are also all of the specific terms provided. And as well as the terms, you get the source details for each term. And so it shows you where this this Go term is coming from and which database assigned the term. And so UniProt is part of the Gene Ontology Consortium, and we exchange data with other consortium members. And so this is really important, so it makes sure that we don't duplicate curation efforts across different resources. So in this case, this particular Go term has been assigned by a UniProt curator, but there are other terms there that have come from other databases, and so they have been assigned by curators at other resources. If you're interested in seeing where in the cell a protein is expressed, then you can go to the subcellular location section, and you get a description of the locations and also a graphical representation, which is provided using Swiss Biopics, which is a freely available library designed to show where proteins are located in cells, and it can also be taken and used in other websites as well. And there's also information provided about expression of particular protein. So links are provided to specialized expression resources. So for example, in this case, you could click on the link to Human Protein Atlas and get a graphical overview of expression patterns for this protein. So this protein, if you're interested in finding out if the protein is involved in a particular disease, you can click on the disease and variant section. So this particular protein is involved in a disease called Charcot-Marie-Tooth disease, which is a peripheral nervous system disorder. And so there's you can find out which diseases are associated with genetic variations in a protein, and the disease-causing variants are provided. So these are the variants that have been extracted from the literature, but if you want to find out, you can combine that with all of the variants that have been imported from large-scale resources by clicking on the Variant Viewer tab. And this will take you to this viewer where you can see all of the variants that are available for this particular protein. You can filter the variants both by the consequence of the variant and also by the provenance. So you can choose, for example, to only see variants that have been reviewed in UniProt, or you could filter for variants coming from large-scale studies, for example. And then there's also a table which provides more information about the variants, the changes, and where they've come from. And this slide shows, so this is the horse ortholog of that human protein. And this entry is in the unreviewed Tremble section. So this hasn't gone through the manual review process, but you can see that the automatic annotation processes we have attached some functional information to the entry, even though it hasn't been manually reviewed. And so this information is coming from the ARBA system. So you can see how, even though an entry may not have been manually curated, there can still be a lot of information provided. And this information is based on the gold standard SwissProt annotation, so is also very high quality. As well as all of the information that is provided within the UniProt database. As I said, UniProt is linked to a large number of other resources, and so you can access information in those other databases by clicking on the external links tab. And so that will take you to the external links section, where all of these cross-references are organized into particular categories to allow you to explore them easily. And this means that you can use UniProt really as a central hub of information, and then move to different resources to explore information that's contained in other more specialized collections. So if you're interested in particular domains or sites within the protein sequence, then you can click on the Family and Domains link in the left menu. And this takes you to the section of the entry which shows important domains and regions within the sequence. You can see these in a table where they're described with their positions and the roles of the domains. And there may also be some textual description to provide some more information about the importance of the domain. And this section also describes the protein family that this particular protein is part of. And important binding sites and active sites are shown within the function section of the entry. And you can see again, you have a table which shows for this protein important binding sites and the active site of the protein. And any sites which have been shown to be post-translationally modified can be found by clicking on the PTM and processing link. So this shows both modified residues coming from the literature and also those that have been imported from large-scale proteomics resources. And again, we have the table showing the residues which have been modified, what the modification is, and the source of the modification. So you can see that using the entry view, these domains and sites are divided into different sections of the entry. But it can also be incredibly useful to view all of these domains and sites and regions of interest within the protein sequence together. And so to do that, you can click on the feature viewer link. And this takes you to the feature viewer, which is a graphical viewer for sequence features. You can see that the features are grouped into particular categories, and you can expand each of those categories for some additional information. So, for example, in this entry, you can see the active site, which is at residue 417. And like I say, it can be very useful to explore all of these features together in a single view. So if you can also expand the variant section, and you can see that there's a variant that occurs at the active site residue. And so this kind of information can be very useful in determining. So this can show, for example, why this particular variant is likely to be deleterious, because obviously disrupting the active site of an enzyme is going to affect its function. And the feature viewer also includes an interactive 3D structure viewer. And if for entries where there is no solved protein structure, then of course we provide the AlphaFold predictions for those proteins. So lastly, I'm just going to show how you can explore the publications that are associated with an entry. So to do this, you can use the Publications tab, which allows you to explore in more detail the literature that's associated with the particular protein. And if you click on this, you get taken to the Publications section, and you can see on the left-hand side of the page that there is a section for papers which have been, so what are called the reviewed papers. So these are papers that have been used as sources of information. So these are the papers that have been read by the UniProt curators and where they've extracted information to add to the entries. And there's also computationally mapped papers. So these are papers which haven't been read by the UniProt curation team, but these are papers that have been imported from other resources and computationally mapped to the entries. And so these are added to allow users to explore more fully the papers that the literature surrounding a particular protein. So if you want to explore the papers that have actually been used during the curation process, you can click on this reviewed link, and this will give you the list of papers which were used during the curation process. And for each paper, you can see what information has been extracted from that paper and why that paper is being cited in the entry. And you can also filter the papers based on the category of data that they provide and also on the study type. So maybe you only want to see small-scale papers and you're not interested in large-scale papers, and if so, you could filter out the large-scale papers so that you can only see those that are from small-scale studies. If you're interested in looking at the computationally mapped papers, then you can click on that link, and these papers are then categorized and the source of each paper is provided. So in this case, this paper has been imported from the Intact resource, which is another resource at the EBI, which collects information about protein interactions. And we also provide the option for users to submit missing papers using the Add a Publication link. So this is what I was talking about earlier with the community submitted publications. So if you are looking at an entry and you want to highlight the fact that it's missing a paper, you can click on this Add a Publication link, and you'll be taken to a submission page where you just need to enter the accession number. In fact, if you click on it from a record, that will already be filled in. And you need to have an ORCID identifier. And then there's a submission form to fill in with the details of the paper. And once they've been submitted, then those community submitted papers appear in the publication section of a UniProt record. So now, in addition to the reviewed and computationally mapped papers, there are also a set of papers here called community maps. So these are papers that have been supplied by the user community. And so any of the data that has been provided is linked to those papers. So in this case, the contributor of this paper also provided some information about the function of the protein reported in that paper. And so that's included. And the publication is also linked to the ORCID ID of the submitter so that they receive some acknowledgement for their contribution. And then the community annotations are shown within the UniProt record as well. So just to summarize, so the UniProt provides a very wide range of functional information. The information is sourced from various places, including the scientific literature, results from sequence analysis, from automatic annotation pipelines, and information imported from other databases. And all of the information is linked back to its original source so that it's clear to users where each piece of data is coming from. And all of the information is made freely available through the UniProt website at uniprot.org. So to access the data, so as I said before, it's released every 8 weeks. The data is provided in a range of formats, and also so the website supports both simple and complex queries, and it also supports programmatic access. So if you're interested in accessing the data programmatically, there's a link to some documentation which can help with that. If you want to follow what's happening in UniProt, we have a blog where we release articles of interest, and then also you can follow us on various social media platforms as well to keep up to date with what's happening in the resource. To access help and documentation, there is a help link in the top right-hand corner of each page, and also this green help button, which occurs halfway down on the right-hand side of the page. If you click on that, then you will be taken to the Help Centre, and there's lots and lots of information and documentation there. So you may find the answer to your question there. But if you don't find what you're looking for, then please do get in touch with us. So we have a very active help desk. So if there's any information that you can't find or anything that you're having difficulty with, then please do contact us, and you'll get a response. So I'd just like to acknowledge the whole UniProt team across the three sites who all work together to contribute to the content of the resource, and to acknowledge our funders who make everything that we do possible. And thank you.
Right, thank you. Thanks a lot, Michelle, for the webinar today, and thanks everyone for joining. We have loads of questions already in, Michelle. So I'll start reading from the top. So first question is, you described this in your talk. What is community literature?
So this is literature that is provided by our user community. So as curators, we spend a lot of time looking for papers ourselves. So like I said, we have various tools that we use to try and keep on top of the literature. But the website also allows users to add publications. So if you find that a particular paper is missing, you can click on that add a publication link that's found on each entry page and provide some details of the paper and what is contained within the paper. And then that paper is included in the record, and the data will be included within the UniProt entry.
How does UniProt ensure the accuracy and reliability of automatically annotated data in Tremble? And are there specific benchmarks or validation steps used?
Yeah, so, well, obviously all of the information is based on the high quality manually curated data that's in the reviewed SwissProt section. And if anything, yeah, so the automatic annotation is constantly, so with each release, it will be refreshed. So if anything changes in any of the underlying data, if any of the signatures change, or if there's changes in any of the SwissProt records, which are part of a particular rule, then the rules will be updated, and so that the data is constantly kept up to date.
What UniProt ID should we take of a particular gene for its protein structure visualization using Chimera software?
So I'm not sure I'm understanding that one correctly, but we always advise using the UniProt accession number. So that is a unique identifier which will never change. So rather than so that we do have these IDs as well, which are subject to change. So the accession number is a stable identifier for each UniProt record.
For the Prot NLM, did you build it from scratch, or is it an open source LLM model retrained for this purpose?
So the model itself has been developed by Google DeepMind. So the model is not actually developed by UniProt. So we've collaborated with them so that we can use this model currently for naming proteins, but I think the plan will be to try and extend it to predict other annotations that we could add to entries. So it's not an open source model, no.
Can you show us how to access unreviewed or uncurated information on UniProt website?
Yes, maybe I can, I could share my screen again. So can you see the UniProt website now? And
Yes.
okay, so if we just use one of the examples that's here, for example. So when you do a search, you can see over on the left bar here, you can choose to see, you can filter for just reviewed entries or for unreviewed entries. So reviewed entries, all the reviewed entries have this gold symbol here to show that they have been manually reviewed, and unreviewed records will have this silver gray symbol to show that they're unreviewed records. And yeah, so when you do a search, you can choose whether you want to see only reviewed or unreviewed. And when you're looking at a particular set of results, anything that has this gold symbol is a reviewed record, and anything with this this gray silver symbol is unreviewed.
How can I use UniProt to determine if specific lipid A modification enzymes, for example, PMRA, PMRC, are conserved across clinical isolates?
I guess the easy thing you could do is run a blast search, and yeah, so the UniProt does provide access to blast. So if you have retrieve a particular entry, you can then launch a blast search and yeah, see what other similar sequences there are within the knowledge base.
How can I use UniProt's cross-references, for example, KEGG, Reactome, to automatically populate metabolic or signaling models in Cytoscape?
That's a good question. And I'm not a heavy user of Cytoscape. So, but UniProt does provide references to KEGG and to Reactome. So the cross references are certainly there. How you can make use of them in Cytoscape, I'm afraid that's beyond my expertise.
How curator give hierarchy and weightage to literature during manual curation?
So yeah, so when we manually curate an entry, we are looking for references that provide novel information. So I mean, there is quite a lot of redundancy in the scientific literature. So we will look for papers that provide novel data. And when we're looking for papers to curate, we're looking for papers that report things like novel functional data, information about new enzyme reactions, information about things like novel splice variants, and particularly if there's any functional information associated with those splice variants, papers reporting post-translational modifications. So all of those kind of things will go towards deciding whether we actually curate a paper or not.
Okay. The next one is, how reliable are unreviewed databases while they can provide you information? Is it a great idea to use that database in your research?
I mean, obviously manual curation is the gold standard, and you know the data quality within the reviewed SwissProt section is considered to be very high. But but also the information that's added to the Tremble unreviewed section is also as good as we can make it. So it's based on the reviewed records which are in SwissProt. It is reviewed and refreshed with each release, and yeah, and only we have cutoff, so only annotation which is above a certain cutoff will be applied. So I guess it depends what you want to do with the data. If you're looking for a very small, reliable set of data, then you can just use SwissProt. If you need a larger set of data, then you can use the unreviewed section as well. And with the caveat that the data is predicted and hasn't been reviewed, but is added with systems which aim to provide high quality predictions.
Thank you. Does deep learning based AlphaFold method can be more reliable than reviewed or unreviewed in 3D protein structure prediction?
I mean, I guess there could be cases where an AlphaFold model is perhaps more accurate than a low resolution structure. So I guess you have to weigh up for an experimental structure, the resolution and things like that, and yeah, to see how you would compare the two.
Does UniProt provide the possible conformations of enzymes and IDPs?
So there is information in UniProt about intrinsically disordered regions, which comes from MobiDB Lite. So there is some information there about intrinsically disordered regions within proteins.
How people manage to review accurately thousands of protein entries so quickly?
Well, I think manual curation process, I would get. I mean, it is a relatively slow process. reading a paper does take time. The team curates somewhere in the order of about 10,000 papers each year. I mean, we have some very good curation tools which help to with things like quickly identifying papers. We also have curation tools which help to speed up actually adding the information to the entries and checking the accuracy of the information. So I guess the sort of the rate limiting step is really extracting the information from the papers, and we're hoping that things like LLMs will help to speed up that process and make the curation even more efficient.
So the next one is, has UniProt this functionality, or is there a link from UniProt platform to predict the effect of point mutation on protein stability or on protein-protein interaction?
Yeah, so there is another resource called ProtVar. So it's not has been developed by UniProt in conjunction with other resources. It's not specifically a UniProt resource, but it looks at the effects of protein variation. So you can go to that resource and you can search for the UniProt accession number and find a lot of information there. So that might be something that you could explore. It might be useful for what you're looking for.